Good morning everyone. This is BHANU SAILAJA KUMARI from Microbiology Department in Andhra Loyola College. Today I am going to explain about ultrastructure of the prokaryotic cell. Prokaryotic is a word come from the Greek language. Pro means absence, karyot means nucleus. Based on the nucleus, the cells are divided into two types, eukaryotes and prokaryotes. Eukaryotes are mainly algae, fungi, protista, plantae, animalia because they have a nucleus and they are multicellular organisms. But in prokaryotes, they have the absence of nucleus, but they are present in the abundant nucleate cells. Nuclear membrane is also present in the prokaryotic cells. The prokaryotic cell is the very small in size. We cannot see our naked eye. We can see with the help of the microscope. So, the first microscope invented by Antony van Leeuwenhoek. That's why we call it as the father of the microbiology was the Antony van Leeuwenhoek. Coming to definition of the prokaryote. The prokaryote is the single cell organisms. We already know prokaryote is the Greek language. Pro means absence, karyot means nucleus. Coming to prokaryote, the size is divided into three parts. The structure will be divided into three parts: external, cell envelope and internal organs. Coming to external appendages, the cells are divided into flagella, fimbriae, pili, glycocalyx, slime layer and capsule. Coming to flagella, the flagella is the consists of a three types of the structural appendages. First one is the filament, hook and basal body. Flagella is mainly involved in the locomotion or movement. The flagella consists of the filament consists of the mainly protein that is called the flagellin. The basal part and hook is encoded with the cell wall, the mainly involved in the movement of the bacterial cell. And next coming to fimbriae. The fimbriae is the very small and hair-like structure. It is the mainly attached to the fimbriae and pilus is the any another bacteria present in the surroundings. They are attract the another bacteria and form a breach and transfer the genetic material one cell to the another cells. We can observe the fimbriae and pili. It is a elongated, ridged and tubular structures. Any bacteria is present in the surroundings there, it is form a breach and they can transfer the genetic material one cell to the another cells. Next coming to glycocalyx. Glycocalyx is the external appendages of the bacterial cells. It is protects the cells in some cases. Next come to slime layer. The slime layer is loosely attached to the outermost layer of the bacterial cell. Its main function is the protect the cell from dehydration and loss of nutrients. Next coming to capsule. Most of the pathogenic bacteria produce the capsule. Its main function is the toxic elements is present. Most of the bacteria is the pathogenic, they can cause us the diseases. The capsule dissolve the toxic element in this capsule, and when we effect of this cause of the capsule, the capsule releases the toxic elements into the human body. Next, the major role of the bacterial cell is the cell wall. Based on the cell wall is mainly two types, gram positive and gram negative bacteria because of the attachment of the peptidoglycan. The peptidoglycan is present in the gram positive bacteria is the outermost layer and it is very thickest. In gram negative bacteria, the outer layer of the peptidoglycan is the very thin. When we using the gram staining, the peptidoglycan is observed the crystal violet and we can appears the purple color. But in gram negative bacteria, when we use the gram staining, when we stain with the safran, the gram negative bacteria is absorb the safran and appears the pink color. So, lipids, proteins and polysaccharides are attached to the cell wall of the gram positive bacteria, but in gram negative bacteria, lipids, polysaccharides, proteins are attached to the above of the peptidoglycan. This is the main important function of the in bacterial cell wall. See, when we observe the picture, the peptidoglycan is the thick layer in the gram positive and peptidoglycan is the thinner this the gram negative bacteria. This is the main differences between the gram positive and negative bacteria. Next, coming to cell membrane. The cell membrane is the look like a fluid mosaic model structure. It is a first observed in S.J. Singer and S.K. Nicholson. It is the main function is the passes the nutrients to the cell and discharges the wastage from the cell. This is the fluid mosaic model because the polar heads are present towards the center. Next, coming to internal organs, cytoplasm and ribosomes, nucleoid and chromosomes and cytoskeleton and endospore. Coming to cytoplasm, dense gelatinous solution is present, it is called the cytoplasm because the prokaryotic cell is the absence of the nucleus. So, the remaining internal organs, that is the nucleoid, chromosomes, ribosomes are float on the cytoplasm only. The cytoplasm main function is the water is the serves to the cells. In water, the mixture of the amino acids, complexes and remaining all the parts are present in the cytoplasm. Because the bacteria cells absence of the nucleus, in the we can has a DNA synthesized into the mRNA. This process is called transcription. This process is done in the only cytoplasm because of the absence of the nucleus. Next, coming to chromosomes. In prokaryotic cells, the prokaryotic cell consists of a 70S ribosomes. In these ribosomes, we have a two subunits are present. One is the largest subunits and another one is the smaller subunits. The largest subunits is the 50S, the smaller subunits is the 30S. These subunits come together and form a cleft, and the mRNA is mainly ribosomes are involved in the protein synthesis. When we form DNA synthesis the mRNA undergoes the translation and to form a protein. This mRNA is undergoes these ribosomes and form a protein. This process is called the translation. In mainly ribosomes consists of three sites: A site, P site and E site. A site is the MNSL site, P site is the pre-exist site, E site is the exist site. So, an mRNA synthesis undergoes these sites and form the polypeptide. This polypeptide is nothing but the proteins. 53 polypeptides form like a chains. This chain is called the protein. Next, coming to nucleoid. Nucleoid because the absence of nucleus, but the prokaryotic cells have a abundant nucleate, that is the chromosomes. Chromosomes are genetic material DNA. The genetic material DNA is the singular and circular chromosomes are present. The extra chromosomal membrane is called the plasmids. The plasmids mainly involved in the modern genetic techniques because antibiotic resistance, vector cloning, they mainly involved in the prokaryotic cells of the plasmids. The best example is the prokaryotic cells is the E. coli. Next, coming to cytoskeleton. The cytoskeleton is a network-like arrangement. The internal bacterial cells which supports the cellular structure. The internal protein polymers is form a closely associated with the cell wall. Next, coming to endospore. Endospore are the dormant bodies. These are produced by the Bacillus, Clostridium, Carcisporsa is the main organism. Most of the pathogenic bacteria produce the endospores. When extreme environment conditions is present in any condition of the bacterial cell, the bacterial cells undergoes the endospore. This process is called the sporulation. This is all about the ultrastructure of the bacteria prokaryotic cells. These are the references books, and not only size, the prokaryotic cells are divided into based on the size and based on the pH and based on the temperature. Based on the temperature, they may be divided into 3 types. They are the thermophilus, mesophilus and saccharophilus. The saccharophilus, the temperature ranges from 0 to 20 degrees, the mesophilus, the temperature ranges from 20 to 40 degrees, and thermophilus, the temperature ranges from 50 to 70 degrees. Based on the pH, they can be divided into halophils, acidophils and alkalophils. Most of the bacteria is the neutrophils. So, the pH will be 0.7 to 8 pH is the present. Based on the size, they may be divided into mainly 3 types: coccus, spirals and bacillus. So, most of the bacteria is the bacilla. Bacillus means rod shaped in structure and they are the spherical and cocci. So, Vibrio cocci is the best example of the cocci of the bacillus. And next coming to oxygen. In based on the oxygen, the bacteria may be divided into 3 types: aerobes, anaerobes and facultative anaerobes. Aerobes means presence of oxygen, anaerobes means absence of oxygen, facultative anaerobes means they may be present in the oxygen, they may be absent in the oxygens. It is mainly useful, facultative anaerobes is mainly useful in the cheese and yogurt and beer and vinegar productions. In industrial purpose, most of the bacteria is very useful. So, the prokaryotic cells is the ultrastructure of a prokaryotic cells. Organelles consists of a mainly 3 types, and cell external organs and cell envelope and internal organs. So, this is all about the organelles of the informations. Thank you.